mentioned in the earlier session, we are going to talk about the actual urine formation, that is physiology of urine formation or physiology of working of kidneys or nephrons. As said, there are three main processes involved in it. The first one is called as glomerular filtration or ultrafiltration. Second one is called as selective reabsorption. Third process is called as tubular or renal secretion. In this session, I'll try to elaborate the process of ultrafiltration or glomerular filtration. Now, broadly, this process is a non-selective filtration process taking place because of a mechanical force, hydrostatic pressure. Now, how does that happen? For that, let's go a little backwards and try to understand what we studied earlier. See, as we saw, the afferent vessel enters the Bowman's capsule, divides into glomerular capillaries, which then reunite and form the efferent vessel. In both the vessels we have identified them as arterial vessels, that is blood contained as a arterial or oxygenated blood. But the point to be noted here is the incoming or afferent vessel is broader, larger in diameter as compared to the efferent vessel. The afferent vessel or afferent renal arteriole is broader than the efferent renal arteriole. And this is one main point in the process of ultrafiltration or glomerular filtration. Second point that's equally important is the role played by filtration membrane. Now, what is filtration membrane? Just have a look here. This is filtration membrane. What we see here is the lining of glomerular capillary, that is endothelial wall. And as we have seen, this endothelial wall has certain pores, certain openings or certain fenestrae. So we have called this as fenestrated endothelium. That's on one side. So blood will be flowing from here. Next to that, this red colored one is the basement membrane on which these podocytes are connected or placed or positioned. Now, the finger-like projections of the podocytes, they create certain gaps, certain spaces, which are called as the filtration slits. Filtration slits. That has another important role to play in the process of ultrafiltration. Now, how does that process take place? See, blood will be flowing in here. And that blood, because of the difference in the diameters of the arteries, that is afferent and efferent arterioles, there's a tremendous pressure developed here. And because of this pressure, because of this hydrostatic pressure, let's say, the fluid part of the blood, when I say I'm majorly talking about the plasma, it starts filtering out into the filtration space. That's a cavity between glomerular capillaries and the visceral layer of Bowman's capsule. That's what we've called as filtration cavity. So this fluid part of the blood starts getting filtered into that filtration space, filtration cavity. Now, what would that be? What would that fluid contain? Basically, these slits, these fenestrae, what we're talking about, they measure to around 2 to 3 nanometers. So, anything of plasma which is smaller than that would simply be filtered out, simply be pushed out. Slightly larger components, they would be pushed out with a pressure, with a force, with some maybe little bit of energy, they have to be forced out. But that too applies to the molecules which are smaller than about 7 to 8 nanometers. The molecules which are larger than this, that is majorly the protein molecules, they will not be filtered out. They would stay in the blood vessel. They would stay in the capillaries. and these proteins would kind of act as a barrier for losing entire water from the plasma. So the blood that is flowing through glomerular capillaries would still contain some water. The protein molecules, they would avoid, they would block the complete loss of water from the blood plasma. But still, almost everything of plasma, that will include water, glucose, amino acids, nitrogenous waste, and basically minerals. All these are going to be filtered out. Now mind well, composition of this filtrate. This is called as a glomerular filtrate, or it is also called as primary urine. Primary urine, which is initially collected in the filtration space, and then it is passed on to the Bowman's cavity, Bowman's space. But the composition of these dissolved in water, composition of filtrate, I am saying, it's almost the same as blood plasma. So concentration of this entire filtrate is almost the same as concentration of plasma. Now at what rate this filtration is taking place? We have identified this as a glomerular filtration rate. As mentioned earlier, every minute around 625 to 650 milliliters of blood would be passing through. Blood, when I say it's majorly plasma, would be passing through kidneys, out of which around 125 ml per minute would be filtered out. This is called as glomerular filtration rate, GFR, 125 ml per minute. Now if we consider this 125 ml of fluid being filtered out every minute, that goes to over 180 liters of fluid. We can't even imagine that much loss of water from our body. So it would be processed and major part of this would be taken back. How would that happen? We will come to understand that in due course of time. But then this 125 ml per minute, it is happening because of what pressure? See, blood pressure in the capillaries, in the glomerular capillaries, is found to be almost the highest in the body. Here the blood pressure is around 55 millimeters of mercury. In the rest of the capillaries, it's around 17 to 18 millimeters of mercury. So it's almost 3 times or a little more. Mind well, that would not be the pressure at which filtration is taking place. Filtration would be taking place because of many other factors. What are they? Let us say this is glomerular filtration rate, that's 125 ml per minute, and pressure that's effective there, it is called as effective filtration pressure or net filtration pressure. How do we calculate it? It's calculated as hydrostatic pressure, the pressure which is developed here because of the difference in diameters and whatever I said, but it is glomerular hydrostatic pressure minus osmotic pressure. See, the fluid contained in the filtration space, it has some concentration which would be offering some resistance to the flow of this filtrate, outflow of the filtrate, minus, that is here it will be plus, hydrostatic pressure in the capsule. Bowman's capsule contains some fluid that also offers some resistance to this. This is around 55 millimeters of mercury. This is around 30 millimeters of mercury. This is around 15 millimeters of mercury, so that is around 55 minus 45, so 10 millimeters of mercury. It's the effective filtration pressure or net filtration pressure. Just try and understand, at this pressure, so much of fluid is filtered out. Just at 10 millimeters of mercury, almost 1/5 of the plasma is filtered out. That is quite an effective process. But then, as you can imagine, all of these are not waste materials. Major amount of water is being drained out, so that will not be lost. Glucose that is required in the body. Same about amino acids, a lot of minerals. So the filtrate that is formed here, the primary urine that is formed here, it's not entirely to be eliminated. Major amount of that, major component of that has to be taken back. And that is what is happening in this second part of the process, that is selective reabsorption. Selective reabsorption. Even the filtration process, it involves a number of controls. First of all, as we saw, the blood pressure here. Now, how is that monitored? How is that changed? We will try and understand that when we talk about regulation of the entire process. Here we have tried to understand just the process of filtration, glomerular filtration or ultrafiltration. How do you like the video? Don't forget to like it, subscribe to it, and share amongst your friends and family. Do post your suggestions or queries. Thank you.